Okay, so our next unit is cellular respiration. So cohort 1 had a little bit of introduction to that this week. Make sure for your virtual Wednesdays that you're taking down the notes. So for cohort 1, you'll present these notes to me either in a written format where you print them out and you write the answers on the on the packet, or you can go ahead and do that virtually where you're bringing a downloaded copy using your computer. So navigate that. If you don't have a printer, obviously bring them in through your computer. Just make sure they're downloaded. The other thing is cohort 2, you're actually going to be ahead where you're going to be taking down the notes and you'll be presenting them December 3rd. Again, that's going to be through the virtual piece where you're using your computer to have the packet done, or you're printing them out. And again, you'll have an open note quiz, cohort 2, based on these notes December 3rd, this Thursday, and cohort 1, that'll be December 7th for you. Okay, so definitely a tough topic. You'll want to make sure that there's our ATP sound, right? Make sure you take your time in understanding this material, okay? And we'll backtrack as we need to in class. So cellular respiration is harvesting chemical energy. That chemical energy, hopefully you know at this point, is going to be ATP, right? Not sugar itself. So here it is. You can see the phosphates. There's 3 phosphates groups. This is actually provides the energy by you actually breaking the bond. It doesn't sound very exciting. You're breaking the bonds. But if you think about what a nuclear bomb is, right, it's actually the breaking of bonds itself. So it would cause an explosion. So it's very powerful, these bonds and the energy that when you actually break them, that's the energy that we're harvesting. So right here is a sugar, believe it or not. It's called ribose. And adenine is nitrogenous base. We'll learn more about those in DNA. So we're going to be comparing contrasting cellular respiration, which is a using of oxygen, in comparison to fermentation, where there is no oxygen used, and how cells will obtain energy from this process. So first off, let's start. What happens if you don't have oxygen? Yeah, I'm sure you can tell by the picture. Without oxygen, we have death. But why is that? So if you said because we get no ATP, you are correct. So how are we going to harvest the energy stored in food? So really what we're doing is we're taking glucose and we're storing it into a different resource, into ATP. That process is called cell respiration. And that food process, or that food itself, is going to be different types. So you'll have proteins, lipids, and carbohydrates. And all that is going to take place in the mitochondria, the making of ATP, right? Not the digestion, right? So the mitochondria is quite small. It's the size of bacteria. And it has 2 membranes, an inner and an outer membrane. And we're going to learn how it actually takes in the oxygen, which we call using oxygen is called aerobic respiration. How it's going to be taking in the food, right, into the mitochondria, and then making our ATP. So usually we use glucose, but you can also use fats and proteins. You're going to always burn sugar first once you run out of your sugar reserves. And don't forget, you have carbohydrates stored in your liver. So you'll burn those first, the sugar, then the carbs in your liver. Then you'll start burning proteins and fats. So this is a chemical formula. Glucose plus oxygen is going to yield you energy, which we know is the form of ATP. And this is supposed to be moved over a little bit, but that's carbon dioxide and water, right? And this is what the formula would look like if it was written with the chemical formula, right? So C6H12O6 plus 6 oxygens give you ATP plus 6 carbon dioxides and 6 waters. For some reason, the PowerPoint, there should be an arrow here. It looks like the PowerPoint didn't save correctly, but. All right, so just like a drill sergeant in biology, we have our little pet peeves. How is cell respiration and breathing different? And how are they related? So respiration is not breathing, hence the 2 different words, right? Respiration is the production of ATP, right, which is cellular energy, which is what we need to keep all our cells alive and, of course, us alive. Breathing is exchange of gases. So if we're talking about organisms like ourselves, we're going to do gas exchange inside our lungs. If we're talking about one-cell organisms, right, their gas exchange will happen at the cell membrane. If you talk about other animals like fish, it'd be at the gills, and amphibians would be through the skin. So how are they related? Well, without breathing, you're not going to have respiration. And again, if you don't have respiration, you're not going to be able to breathe. So they go hand in hand with that. Breathing provides the gas exchange in order for respiration to take place. So what do we need to make energy? So first of all, we need our mitochondria, right? They're going to be found at different concentrations in different cells. So the more energy you need, the more mitochondria you're going to have in your cells within reason, right? We need to build up a stamina. So we're going to build up mitochondria. Remember, mitochondria can actually reproduce. They have their own DNA inside. So the more energy you require of your cells, the more mitochondria are going to be produced, and so you'll have an increase in stamina. Then we're going to need our fuel source. The fuel source usually is going to be carbohydrates, but again, as a backup, you have fats and proteins, right? So food overall, we're really going to be focusing on glucose as a main energy component here. Helpers, so we definitely need our enzymes, right? We need our oxygen in order for that cell process to take place, which is the production of ATP. As a byproduct, we also have our wastes, right? And that waste would be the formation of carbon dioxide and also water. So even though we call carbon dioxide a waste, it really is used by plants in order for photosynthesis to take place. So what is a waste to us is value to other organisms. So, but in this process, we do call it a waste product because we ourselves are not using the CO2. Okay, so this is what's going in. These are your inputs, right? Remember, talked about chemical reactions, kind of like baking, like this is what you're adding. This is the manufacturer, right? This is your furnace, and then it's outputting your ATP, and as a byproduct, the CO2 and the O2. Right? Look, this is what the mitochondria are saying right now. I make ATP, all I do all day, and no one even notices, right? But hopefully now you do. See how important they are. So for some reason, they don't think that you get this on the regions. They want you to say mitochondria are only found in animal cells. So only animal cells need to make ATP? No, that happens on the cellular level. So an amoeba has mitochondria, a lion has a mitochondria, plants have mitochondria. Every cell has mitochondria, except there's always an exception, bacteria. Bacteria are actually going to make their own ATP through their own cell. So you got to remember bacteria are the same size as mitochondria. So, and you got to remember mitochondria are bacteria themselves, right? They have their own DNA to replicate. They're endosymbionts. So mitochondria are found everywhere in animal, plant cells, in any type of eukaryotic cell. So that would be like an amoeba, paramecium, but not in prokaryotes and bacteria. Bacteria, like I said, are the size of the mitochondria. Okay, and of course you can find those cell organelles found in the pictures there, right? So using ATP to do work, we can't store ATP. It's very unstable. It has high energy bonds, and we use those energy bonds to create movement in every one of our chemical reactions in our cells. It's very quick, short time energy. We'll see that in the next slide. And then what we do in order to keep ourselves going is we store this extra fuel, right? The good thing about food is that it's a very stable component. It's a polymer. It's very stable, can last for quite some time. So you store it in your body. It could be your whole lifetime, like in fats, right? Or carbohydrates are going to be shorter term storage. So a couple of days, a week maybe for that carb storage, if you're not replacing it, right? So we know it as ATP as adenosine triphosphate. So there's 3 phosphates here, right? And it's actually the phosphate that pulls off that gives you the energy, right? The breaking of that bond. And that's going to provide work. That energy is going to provide work to any type of cellular activity. So what happens with ATP is once it uses that extra phosphate, that phosphate still stays inside the cell, but it's now reduced down to adenosine diphosphate. Sometimes in extreme situations, you can actually get adenosine monophosphate, but your body is extremely under stress in order to give it energy if that's the case. It's very, very rare for that to ever happen. And then this is all taking place in the mitochondria, right? So just to give you an example, a working muscle recycles over 10 million ATPs per second. Right, that's how quick this reaction is happening. So ADP, that phosphate's still floating inside the mitochondria, and then we're going to learn how that phosphate gets slammed on to ADP to bring it back to ATP. And again, this is all taking place in the mitochondria. Okay, so just kind of give you an idea about that short energy determinant here. If you look, this is your overall performance. Okay, so really what it's saying is that you have 1 minute of storage of ATP. So if you look right here, you have your ATP store. Well, about 2 seconds worth. But you also have an ATP system. We're not going to get into that. But either way, you can see it right here. And then we actually have now, let's say you don't have any oxygen, you have something called anaerobic respiration. So you really only have about 1 minute of stores just to rely on your cellular activity where there's no, let's say there's no oxygen, right? So if you need stores of ATP to keep your body going, you have about 1 minute worth. Or even like if you're doing exercise, you have bursts of energy, and that burst of energy sometimes is actually extremely quick, maybe only a couple seconds, but 1 minute overall. To keep that, well, I shouldn't say sustained. Honestly, you only have right here is a threshold. So maybe a couple seconds of a burst, and then it starts diminishing, right? About a minute. So to keep ourselves going after a minute, that's kind of what life is all about, surviving longer than that, is that you do something called aerobic respiration. So if you need to keep that living thing going, you're going to do aerobic respiration. You can see how it's going to be continuous, and that's going to give you overall performance. So sometimes we get that energy, right? It could be adrenaline, but it also is that ATP stores maybe are going to be providing that burst of energy if it's needed. So our body's energy budget, how do we spend that energy, right? So let's say we're eating our food, we're eating our proteins, we're eating our fats, we're eating our carbs. That's going to give you energy, right? We know that energy is a source of ATP. So you're going to even need it if you're just sitting there, not doing anything, just sleeping all day, breathing. You need to, keep your temperature at 98.6. You need to keep breathing, right? So all that's actually going to require some energy stores, right? The other thing is when you're eating food, you're obviously not just going to be making energy from it. You're also going to be building things. So you're going to be taking those, amino acids, and you're going to be making your own proteins. Maybe you're taking those carbs, and you're not using them all for energy. You're storing some of them later on as starch. So you're going to be using that later on for growth and repair of your own body. All right, and then the ATP also could be used not just for building, right, but also for storage. So that storage is usually in animal starch or fat. You usually don't store proteins. So really, you can utilize proteins in, like they call it, a ketogenic diet. But for the most part, you usually go into starch and then you go right into fat reserves. So you have to burn those carbs first before you get to burn those fat calories. So that's sometimes a hard weight loss for people, is that they might eat less calories, but if they're not burning that fat, then they won't necessarily see a reduction in their fat content. So that's kind of a tough thing for people when they're learning how to lose weight. So here's a simple formula, which even looks hard enough. So we have our first stage. Glycolysis is an anaerobic stage. Okay, so this is where no oxygen is actually required. Okay. The last two stages are going to be aerobic. So let me just go back real quick. If you go back, two slides. All right, so here you can actually see your anaerobic respiration. This is going to be, after that's going to be aerobic respiration. So we need that respiration, that oxygen respiration to keep ourselves going. But we do have a little bit of time where we're not breathing, right? Or not breathing, that we're not using that oxygen. So we have a short period of time where we can do anaerobic respiration. All right, so this is a summary equation. We have our sugar plus our oxygen. That arrow means yield. And you should know that we need enzymes in order to do any chemical reaction, right? So we'll definitely talk a little bit about enzymes. The good thing is you don't have to memorize all the enzyme names. And then we have our carbon dioxide and our water as waste. And of course, we have multiple ATPs being made through that process. Here comes the hard part. So respiration occurs in three main stages. You have one, glycolysis, two, Krebs cycle, also known as the citric acid cycle, and then finally our electron transport chain, also known as oxidative phosphorylation. So that's a mouthful. I'm going to always really refer to it as ETC. And when you get into chemistry or AP Bio, you will learn it as oxidative phosphorylation. All right, so this is the initial fuel source. We're going to keep it simple. We're going to use glucose. It could be proteins or fats, but we're just going to use glucose. Here's our first step, glycolysis. Our TCA, which is the citric acid cycle, and then our electron transport chain. And this is going to give us our ATP. This is all aerobic cellular respiration. So going back, right, this whole process is what we're going to go into, but remember, it has three steps. Okay, so even though I said one chemical reaction, there's actually three steps to that chemical reaction. All right, and it gets a little bit more confusing. All right, so what we're going to do here is, oops, sorry about that. We're going to have to label this diagram in class, unless you can handle it. Maybe try it. But in class, we'll definitely go over this, this process of glycolysis. So glycolysis means to split sugar. It's a series of chemical reactions that happens in the cytoplasm, not the mitochondria. You do not need oxygen. It's actually an anaerobic. So you have a little bit of stores of energy. You make a little bit of ATP, but not long, maybe a couple seconds worth, right? And so you're going to be making pyruvic acid behind glycolysis and a little bit of ATP. So here it produces only 2 ATP molecules for each glucose molecule. So it's broken down to the net amount of 2. So you actually have to put 2 ATP to get this chemical reaction to even take place, right? And then what's going to happen is you're going to make 4 ATP. So 4 minus 2, let's not deal with negatives. So 4 minus 2, you put 2 in, and you're making 4, your net or your overall amount that you're making is only 2. So you're making 2 ATP. You also have something called NADH that's formed. And NADH, we're going to learn later on, is really important in carrying hydrogens. Okay, so we're going to have to make a concentration gradient, right? Remember that difference between the plus and the minus is going to make a dam, right? Like a waterfall. And that's going to provide the energy, that movement to allow molecules to move across those concentration gradients and to spin something called an ATP synthase. So we'll get into that in a little bit. So we're going to do this in class just to make sure that you understand it. The net ATP is 2. If you want to tackle that now and write in pencil, maybe give it a shot. And then when we come to class, you'll have a little bit more, maybe you'll be able to contribute some answers. So here, we're going to have 2 ATP going in. And then this 1, 2, 3, 4, 5, 6 carbon sugar is going to be called glucose. I know it's broken down. It's not a circle, which is normally how glucose would appear, but 6 carbon sugar, right? And what's going to happen is you're going to actually take ADP, right? You're going to take those 2 ATP. You're going to be using that energy. So you're putting in 2 ATP, right? And from there, you're going to be making 4 ATP, right? So again, the net would be 2. You're also going to be making glucose is now going to be making 2 pyruvic acids, which you can see are 3 carbon sugars, if you want to call them that. And then you're also going to be making 4 ATPs, which are right here, right? And also what you're going to be doing is you're going to be taking your NAD plus another phosphate or energy requirement from the ADP. So you're using these phosphates in order to make NAD into NADH, which is a hydrogen carrier. Okay, so here you have your 2 NADAs, 2 H's plus 2 NAD pluses, right? So where would we label that? All right, that would be over here. Okay, our 2 ADP, right, and our 2 ATP, right, going in. Okay. So how did glucose get into the cell for a heterotroph or an autotroph, right? So if you're an autotroph, how did you get sugar? If you said made it, perfect. If you said heterotroph, they have to consume it, correct. So the whole point behind plants and making sugar is so they can use it in their own mitochondria to make their own energy. So when we look at our glucose, we actually see glucose goes into the cell, right? It's very small, can fit through the cell. And here's the mitochondria kind of blown up. And glucose is actually broken down into 2 3-carbon sugars called pyruvate. Now, if there's no oxygen, we'll talk about this later, ethanol or lactic acid is made. If oxygen is present, then it's going to go through the process of aerobic respiration. This process right here of glucose being split into 2 pyruvates is called glycolysis. That's the splitting of sugar. So there's actually a transitional phase before this whole process. It's actually the making of acetyl-CoA. So before the citric acid cycle, which is right in here, before we get to that step. So yes, we said glycolysis takes place. You have your 2 3-carbon sugars called pyruvate or pyruvic acid. Before pyruvate goes in to the mitochondria, it becomes acetyl-CoA. It actually forms something called acetyl-CoA. So before the citric acid cycle, which is here, and then before the electron transport chain, which is the third step, pyruvic acid undergoes a series of chemical reactions, which gives off a molecule of CO2 and actually combines with a molecule called coenzyme A to form acetyl-CoA. Acetyl-CoA winds up being 2 carbon sugars. And one of those carbons is released as CO2 as a byproduct. So here we see our pyruvic acid. One is coming off as carbon dioxide, which is a waste product. We actually have an enzyme, coenzyme A, actually vitamin A. That's why vitamin A is so important. Helps with cell respiration. And then you have an enzyme that actually takes that 2-carbon sugar, which you have over here, and attaches to CoA, coenzyme A, which is vitamin A, becomes acetyl-CoA. So this whole thing is called acetyl-CoA. So it's vitamin A with 2 carbons on it, or 2-carbon sugar, right? That is actually going to go in and spin through the citric acid cycle. All right, you're right. So first we have glycolysis, which makes pyruvate. That pyruvate is going to now get changed to acetyl-CoA. All right, so it doesn't get any easier. So now we have the citric acid cycle. So here's our pyruvate. Here's our acetyl-CoA being made. And now we're talking about the Krebs cycle. So there's a quick animation that we'll go over real quick in the next slide. So pyruvic acid from glycolysis combines with coenzyme A, which is vitamin A, to become acetyl-CoA. That's a transitional phase. So pretty much verbally what I was saying before is written up here, okay? So here you have that. And remember, you have a carbon dioxide coming off of it. You also have NADA being oxidized into NADH, which is hydrogens. This is going to come later. These don't seem that important because we're really just focusing like on ATP and things like that, but these you'll actually see will be really important. Acetyl-CoA enters the Krebs cycle. Each turn of the Krebs cycle produces 1 molecule of ATP and FADH plus 3 molecules of NADH. So really what's happening is, if you look here and you're writing this down, the Krebs cycle's whole purpose is actually making this stuff. So yeah, you see ATP, but that's not the whole point behind it. So for glycolysis, you make 2 ATP. The Krebs cycle, if I remember right, I think it's 2 ATP as well. But you'll be making, it's 2 ATP, sorry. You'll be making 3 NADHs and 1 FADH. Now this is for 1 pyruvate, so you got to do a second spin. So you got to multiply that all by 2, okay? So instead of 3, you end up having 6. Instead of 1, you have 2. Who cares, right? What we were talking about ATP now, who cares about NADH and FADH too? We're going to need those for hydrogen to set up that concentration gradient, right? It's always, think about your car battery, right? You got a positive and a negative charge. You have to do the same thing here. You have to create a difference in order to get energy, right? The dam, the water's higher on one side and lower on the other. Let the water fall and push a turbine, and that's how you get electricity. So that's what our body is doing at the cellular level. Okay. Tough stuff, I know. We're going to review this, but we're going to review this in class. We're going to be looking at how pyruvic acid, pretty much everything in this picture is a quick animation. So you'll be hearing it, writing notes, let it sit in, and then in class we'll be going over that as well, okay? So here's our final stage, almost. We have anaerobic respiration, but that's not as bad. The electron transport chain occurs in the inner mitochondria. So where's the inner mitochondria? I think I might not have a picture here. So here, if you look at the mitochondria, there's these little folds. Inside the folds is the inner mitochondria, okay? So this is where our ADP is going to be made, where another phosphate is going to be added to make ATP. So electron transport chain, that's where the ATP is made. So the ETC, which is abbreviated as electron transport chain, is a collection of carrier molecules. So these molecules are all embedded inside this membrane. So like right in here would be an example of where they'd be found, right? Not up here. Okay, that would be the outer membrane. So it's the inner membrane. So what it is there's all these little circles and things like that. They're proteins. What is their job? Pumping. Okay, what are they pumping? Those hydrogen ions. Where do we get those hydrogen ions? From NADH and FADH2. So big deal. We have hydrogen ions. What do? Create a concentration gradient. And what they're going to do is you're going to have a lot of H's on one side and not a lot on the other. So oxygen is a final electron acceptor. It's actually going to pull those hydrogens in, and it's going to create a spinning effect on the very, very last protein carrier molecule, which is called ATP synthase. And that's actually a turbine that slams into another phosphate to make ATP. The net amount is approximate. It's different for each person. It's kind of an average. It's 36 ATP. for every sugar molecule. All right, so we sometimes will say like the net amount is 36 ATP, but even sometimes 38 based on glycolysis and also the Krebs. Again, no one's going to ask you exactly how much ATP is made. The magic number sometimes is 36 based on this, piece found in a textbook, but it's not an exact science yet. All right, so the video just shows you, it's a GIF animation, really what's happening inside the inner membrane. So here, try to show you a little bit better with the mitochondria. So there's an outer membrane, right? So here's like the outer part. Then there's these little pieces inside the mitochondria called cristae, and they actually form the inner membrane. So there's these folds. So this is happening inside that spot. Remember, glycolysis is happening in the cytoplasm. That's without oxygen, and all of the citric acid cycle is happening in the inner mitochondria. And then this is happening actually like in this part, right? So these little folds right here. Okay, so what's happening is you're getting all these hydrogen protons. Those hydrogen protons were carried by NADH and FADH2. It's not shown in this animation, but they're pushed onto one side. So you're getting this concentration gradient. So they want to diffuse back in. So there's these carrier proteins, and it doesn't show here in the animation, but they actually carry electrons all the way through. That electron is negative, and what's going to happen is those hydrogens are going to want to even go further because those electrons are negative, and they're going to slam into, here it is. So this is the thing that's highlighted. It's called ATP synthase. It actually looks like a turbine. It legit looks like a molecular turbine. And it takes the ADP, it slaps onto another phosphate, and then it goes ahead, and then that ATP gets used for cellular energy. So it doesn't show you here the best, but what's attracting it is oxygen, right? So if oxygen wasn't here, O actually would wind up being negative. If the negative oxygen wasn't here, the hydrogen would never want to pull through so fast. So we call oxygen the final electron acceptor, and that attracts the other part, as well as being a different concentration gradient. So a really quick review, we have our glucose, which is C6H12O6, being broken down to 2 pyruvate molecules, which is going to be only 3 carbons. So basically it's digested. Sugar is broken down to 2. This is all happening in the cytoplasm, which is also the cytosol. And you're going to be taking that pyruvic acid now, and you're going to change it. You're going to be removing one of the carbon dioxides off, one of the carbons off to make carbon dioxide, and you're going to be putting on vitamin A on there. So you're going to wind up with something called acetyl-CoA. So it'll be 2 carbons with vitamin A attached. That's acetyl-CoA. Remember, that came from pyruvic acid. So that's actually going to go through the Krebs cycle. You're going to be making a little bit more ATP through the Krebs cycle, but our whole point behind the Krebs cycle is to make FADH2 and NADH. So here they show you the numbers here, right? Because you remember, you got to do this turn twice because you have 2 carbon, 2 pyruvic acid. So they're showing you the total here. Then here's the main part, electron transport chain with the oxygen. Here's O2 is actually going to make a little bit of water as a byproduct, but it's going to give you approximate ATP. So 4 ATP, because they're saying 2 from glycolysis and 2 from Krebs cycle, and approximately between 20 and 30 made from the electron transport chain by spinning that turbine, that ATP synthase turbine. Again, it's approximate. The magic number tends to be in most textbooks 36. Okay. There's another pathway. This one's a little bit easier. This is the easy pathway, right? It's one direction. So if you don't have any oxygen, whether it be your own personal body, or if you're, like we looked at the yeast, if there's no oxygen, they're going to do something called fermentation. So if oxygen is missing, so move my head, you can see no O2, okay? You can't complete aerobic respiration. That makes sense, right? So yeast are going to do this all the time. Bacteria are going to do this all the time, and it's called anaerobic respiration. We also know it commonly as fermentation. Also alcohol fermentation, there's 2 different types, and lactic acid fermentation. That's the one our bodies do. So again, if you're a mitochondria, that's what you have to do. Anaerobic. You actually can do aerobic respiration, but you can perform anaerobic as well as bacteria. And it's a very small amount of ATP, but if you're only 1 cell big, you don't really need a lot of energy. So 2 ATP for anaerobic respiration is good. If you're large, not going to work so long, right? We don't have very long with anaerobic respiration. So fermentation for yeast is going to do alcohol fermentation. So make beer, wine, we can make bread, right? And again, you're going to be taking that glucose, no oxygen, right? And you're going to make ATP, CO2, and alcohol. There's another process called lactic acid fermentation. So bacteria do this and animals do this. So it's where we take glucose and we're going to be, again, no oxygen. We're going to make ATP. So why do we do that? We'll get into that. So bacteria are going to make us yogurt. There's other products that they make too. I'm just not remembering it. But bacteria are going to ferment. They're going to be making different products for us. Like I think certain, not tofu, but there's certain fermented products that other people use besides just like yogurt and sour creams and things like that. The other thing is that we have our muscle fatigue from that. And so you can feel that when that's burning your body, that's lactic acid burnout. So there's a state lab. I think I might save this for later on so that if we do have the regents in June, and like we'll do this like in May. So it's a little bit more, a piece that you would remember for the regents. It's kind of an easy lab. Really what it is showing that energy requirement that you need, and then you actually have a lactic acid buildup. So we get to exercise in class, and we're going to measure, our weakness and strengths behind lactic acid fermentation. So I'm sure you can assume you understand that we can only do it for a certain amount of time without oxygen, and then we run out of steam, right? It actually burns a little bit. So here you have your pyruvic acid. You're going to be getting the same thing, which is glycolysis. But here it's going to take a little bit of a different pathway if there's no oxygen. So lactic acid would be made, or fermentation if we're talking about alcohol fermentation. So in humans during strenuous exercise, if there's not enough oxygen, so even though you're breathing, you're just not getting enough oxygen to all your muscle, you go into oxygen debt, and then you start accumulating this lactic acid in the muscle. So it actually leads to like fatigue, pain, but again, your body will, once you start catch your breath, will regain itself. Alcoholic fermentation is very similar, where you're taking the glycolysis step, but yeast happen to do this. And they're going to go ahead and they're going to take that pyruvic acid. They're going to still be making a little bit of energy, but they're going to be making ethyl alcohol with carbon dioxide. So that's kind of the carbonation process that we see in the alcohol products, right? Or if we're making bread. All right, so we're going to try this game. If you want to try it now, I don't know if you can with the YouTube video, but you can with your note packet, so you can try it. Maybe we'll have a little contest in class. All right, talk to you soon, guys.